In this slide, we are going to talk about meiosis. And meiosis is a very unique kind of nuclear division that occurs only in the gonads. And it's the same process for males and females. So we refer to this also as what is called gametogenesis. And gametogenesis is a general term that refers to both spermatogenesis and oogenesis. So sexual reproduction cannot take place without meiosis occurring because the most important thing is that it reduces the number of chromosomes in the gametes by half. So the zygote does not end up with twice as many chromosomes. So for example, the sperm is going to have 23 chromosomes, and the ova is also going to have 23 chromosomes. So when fertilization happens, there's now going to be 46 chromosomes in the zygote, and the terminology for that is going to be 2N. So some of the terminology in meiosis is that of sister chromatids. And what that is it's the cell's DNA that's replicated during interphase. Each chromosome has two sister chromatids that are identical and joined together by a centromere. The DNA coils and condenses to make chromosomes visible when viewed through a light microscope. So to give you an example of kind of what this is going to look like, if we have one chromosome here, we would have a centromere in the middle of it. And each of these regions would be a chromatid. And all along that region of chromosome, there are DNA segments which are going to encode for a specific trait. And those are, there are homologous pairs of chromosomes. So one member is from the father, the paternal chromosome. Another is from the mother, which is the maternal chromosome. So they look alike and they carry the genes that code for the same trait. So what we're going to talk about more, especially in chapter 29, is some of the stuff that you've probably heard before. So each of these traits are usually represented by a letter. There's a capital letter and a lowercase letter. Let's say in this case, and it's random as far as what exactly the trait is. So if there's a capital B, that could be for brown eyes. Since it's the capital letter, it's the dominant trait. Blue eyes would be the lowercase letter. So when we would see something like this, meaning that the paternal trait could be brown eyes and maternal could be blue eyes, this person would have brown eyes because the brown trait is dominant to the blue trait. If the person got two blue traits from the mother and father, then that child would have blue eyes. This is also represented on sex chromosomes. So I'm sure you've heard of sex-linked traits, and these sex-linked traits are going to be associated with either the X or the Y. So we have autosomes, and those autosomes are non-sex chromosomes. So there would be 22 pairs, which would be 44 chromosomes. The remaining two would be sex chromosomes. And these sex chromosomes are one pair. So the one pair is the additional two chromosomes. And those particular chromosomes could be one of two, obviously. The person could be XX or they could be XY. And they discovered that the Y chromosome is actually smaller than the X chromosome. But a sex-linked trait occurs when something is represented only on the X, and if there's a male, there's not going to be another X allele to sort of cancel it out. So in this slide, for meiosis, the terminology, we have haploid, which I've already mentioned to you. So we have the number N representing the number of chromosomes in the gamete. The gametes have only one member of each homologous pair, either maternal or paternal. So the haploid chromosome number is 23. When the sperm and egg fuse, they form a diploid, which has two N chromosomes. So again, remember that 2N equals 46. So for the sequence of events of meiosis, it's different from mitosis because it has two consecutive nuclear divisions, referred to as meiosis 1 and meiosis 2. The product is 4 haploid daughter cells. So each nuclear division has the 4 steps, which are prophase, metaphase, anaphase, and telophase. And you learn the same names for mitosis in A and P1, but now we have 2 stages of this. So we still have the PMAT. To me, it sounds somewhat like a standardized test, like the GRE or the MCAT or the NCLEX, something like that. So if you remember PMAT, that will help you remember the names of meiosis in order. So there's prophase 1, metaphase 1, anaphase 1, and telophase 1. The most significant phase of meiosis 1 is prophase 1 because there are mixing up of the genes. And we'll talk about the mechanism of how that works coming up. So the star here means mixing up the genes. And again, the most significant part of meiosis 1 is going to be that prophase 1. So here we see that we start with diploid. This is meiosis 1. After meiosis 1, the chromosomes have been reduced to haploid. Then we have meiosis 2. So we have another PMAT, but it's prophase 2, metaphase 2, anaphase 2, and telophase 2. So in this situation, each of those haploid are going to be copied into other haploid daughter cells. So the end result is a total of 4 daughter cells. So this is meiosis 2, and it's like mitosis. And it's important to remember because mitosis is an exact copying of the chromosomal material.